welcome to ACR Journals on Air podcast. I'm your host, Vicky Shamagam. During training, most rheumatologists learn that 90% of the diagnosis comes from the history. But I wonder how many of us use history to learn immunology. The manuscript we will discuss today is Standing on Shoulders, Interferon Research from Viral Interference to Lupus Pathogenesis and Treatment. It was published in Arthritis and Rheumatology as part of the new Immunology for the Rheumatologist series, and a link to the manuscript and disclosures are in our show notes from today. I'm thrilled to be joined by the manuscript's first author, Dr. Peggy Crow. Dr. Crow is professor of medicine at Weill Cornell Medicine and professor of immunology in its Graduate School of Medical Sciences. She is the chair of immunology and inflammation research, senior scientist, and co-director of the Mary Kirkland Center for Lupus Research at Hospital for Special Surgery in New York. Dr. Crow received her rheumatology training at Hospital for Special Surgery and postdoctoral research training at Rockefeller University. Dr. Crow's research has focused on the induction and regulation of human autoimmune diseases, and she has devoted her career to studying the cellular and cytokine mediators of immune system activation and inflammation. Dr. Crow identified type 1 interferon as a critical molecule involved in the pathogenesis of systemic lupus erythematosus, and the mechanisms of interferon induction in lupus pathogenesis is a significant focus of her work. Dr. Crow is also a leader in the field of rheumatology. She is a former president of the American College of Rheumatology, president of the Henry Kunkel Society, chair of the Scientific Advisory Board of the Lupus Research Alliance, and chair of the Medical and Scientific Advisory Council of the Arthritis Foundation. From 2010 to 2020, she served as physician-in-chief for the Hospital for Special Surgery. In 2017, Dr. Crow was named as an honorary member of the European League Against Rheumatism. And in 2018, in recognition of outstanding contributions to rheumatology over the course of her career, Dr. Crow received the ACR Presidential Gold Medal. In 2022, she also received the Lee C. Howley Senior Prize for Arthritis Scientific Research from the Arthritis Foundation. Welcome to the show, Peggy.
Hi, Vicky. It's nice to join you.
So many of our listeners know that history is incredibly important in making a diagnosis, but it was a real treat to read your manuscript, which is a beautiful history of interferon research. Not many scientific articles are written in this way. What made you focus on the history of the interferon story in this review?
Well, it is a bit of an unusual way to approach writing a review, but I think it really stems from my great respect for researchers and what it takes to build, the fund of knowledge that, can lead over time to really important observations. I've been a researcher for many, many years, and, you wouldn't believe what we didn't know 40 and 45 years ago. So I've worked on interferon a long time, and over that period, I've thought a lot about the early investigators in this field and what they've contributed, and some who've gotten a lot of credit, some who've received less credit. So to really answer your question, I was stalling on writing a review on interferon that I'd been asked to write for A&R. And finally, in a discussion with Rich Fury, who's one of the associate editors, we worked out a plan that it would be okay for me to focus on the history. So that's what I did.
Well, I'm so glad you did, because I think it's a wonderful manuscript. So the beginning of the interferon story is really interesting because it starts with the discovery that in viral infections, it is relatively unusual to be infected with two viral infections simultaneously. And several investigators were working in the field, including a group from Japan and the investigators Isaacs and Lindemann, who are now thought to be the founders of cytokine biology. Tell us about their work.
Right. Well, there was great interest back in the 1950s in understanding how, in the setting of an infection with a virus, the immune response could try to battle that virus. As you said, Isaacs and Lindemann have received great recognition as really the fathers of interferon biology. But, looking in more detail into this story, I found that there were two Japanese investigators, Nagano and Kojima, who were also studying the response to virus infection. And they've probably received a little less recognition. And, it's interesting to think about why that might be. They were working in a rabbit model, a rabbit skin model of vaccinia virus infection. And they did recognize that if they administered inactivated vaccinia virus to these rabbits, and then within a reasonably short period of time, infected the rabbits with a second virus, they were protected. But it was the nature of dealing with rabbits and skin that they really couldn't get a grip on, how this was working. So it was really when Isaacs and Lindenman, Isaacs, the key investigator, with Lindenman, a visiting young student, really, using an in vitro system, that they were able to detect a substance in supernatants of cell cultures that represented this inhibitory activity.
I love that. And I feel like that was something that was throughout your manuscript, this concept of, the biology is always telling us something, and sometimes we're just waiting for the science to catch up.
Yeah.
So tell us next about Jan Wilczek. He started publishing on interferon in 1960. Was he the first person to call it interferon? And how did his work advance this field?
Well, it was really Isaacs and Lindemann who called interferon. In fact, their famous paper was called the interferon. and so they coined the term. But I found Jan Wilczek so interesting, and perhaps particularly interesting because he's still a presence in the New York area. He's been at NYU for many, many years. But Jan Wilczek started his lab as a solo investigator in 1957 in Bratislava, Czechoslovakia, a communist country at the time. So he was straight out of medical school, interested in virus infection. And very much like Isaacs and Lindenman, he used an in vitro system to study protection conferred by virus infection. And he was able to take supernatants from cultures and for years really dug into what this inhibitory substance was. And he continued his work leaving Czechoslovakia, moving in 1965 to NYU, where, as I said, he remains as a senior investigator. So over the years, he characterized interferon as a protein substance and characterized actually several types of interferon, and then worked with many other investigators digging into exactly what this substance was. One of the key observations was that there were at least two types of interferon, what he called leukocyte interferon, which ended up being called interferon alpha, and fibroblast interferon, which is interferon beta. But he made many, many contributions on his own initially, but then through collaboration with many wonderful investigators.
Yeah, and as this work was being done, as I'd mentioned, the scientific methods were evolving too, and investigators began to eventually sequence the genes. It sounds easy now, looking back, but the work really required a community of scientists, each building on prior findings. What can you share with us about that part of the story? And what do we now know about the differences and similarities between the different interferon molecules?
Well, there was a point in time, 1980, when the competition among investigators, including pharmaceutical companies, to clone and define interferon was fierce. It's very interesting that there was such activity and so many publications in that one year. And you're right that it really required technology to evolve so that investigators were able to clone genes that enabled this real flurry of activity. But I'll just also mention that kind of in the background of this competitive activity was the thought that maybe interferon could act as an anti-tumor agent. There had been some mouse studies suggesting at least an anti-tumor effect of interferon. So many companies thought, well, this could be an anti-cancer drug.
Mm-hmm.
So with the availability of cloning, though, Nagata, Charles Weissman, Taniguchi, David Goodell at Genentech, all were involved in cloning both interferon alpha and interferon beta. In the case of alpha, some of the investigators went on to define in detail, a number of different genes that encode a number of different protein products of interferon alpha and only one protein product of interferon beta. Now, you asked, what's the difference? And I think that from my point of view, I think there's still a lot of questions about what's different, and we may get to this. They both bind to the same receptor, but they, have some modest sequence differences. And as I mentioned, they're predominantly made by different cell types. So some differences, but they all end up acting on a pathway that, we've learned is very important, particularly in antivirus host defense, but, also in a number of autoimmune diseases.
Yeah. And so let's get to that. So, one of the main functions of type 1 interferon, as we've talked about, is to mediate the response to viral infection. But it does also have these other roles in augmenting other immune responses. Tell us a little bit about that. And then what does this term priming mean?
Right. Yeah. Well, as you can imagine, for a system that's really essential in combating virus infection, type 1 interferon, and type 1 interferon is kind of the global term for interferon alpha and beta, and now we know there are others, kappa, omega, and so on. Many functions are impacted by type 1 interferon. I think the first function that was observed was the activation of natural killer cells. And we actually our first interferon related paper was studying the effect of type 1 interferon on NK cells in the human system. But there's so many different activities, and I think that we continue to gain understanding of how type 1 interferon impacts the immune system. And it augments B-cell differentiation, it augments antigen-presenting cell function, it affects T-cell function. So, it really orchestrates how the immune response can behave when it's faced with a virus infection. Now, you asked about priming. Priming is a term that has kind of shifted into what's now called training, but I think either term is very appropriate. It really suggests that if a cell experiences a small amount of type 1 interferon, and there's some data that suggests that all of us have kind of a low, smoldering level of type 1 interferon all the time, that cell is then trained or primed to respond more robustly when exposed to a second stimulus or a second amount of interferon, or it could be another kind of stimulus. And what priming or training really reflects, as is being understood now, are epigenetic modifications to DNA. So a little bit of interferon is experienced by the cell, and the chromatin is modified, whether through changes in methylation or acetylation of histone proteins, in a way that the cell is more ready to respond on a subsequent trigger. So that's what training or priming means, and it's really an area of great interest now to investigators.
Yeah, I'm sure, and particularly in regards to autoimmune diseases, I'm sure it's an area that's ripe for research. So tell us now about the interferon receptor. How is this described, and what is the relationship between receptor affinity and actual interferon response?
Well, the first description of the interferon receptor came from the lab of an investigator named Frank Ruddle, and he did just the most fascinating kind of experiment. He generated cell lines, each of which expressed one human chromosome. And then these cell lines, each expressing a human chromosome so it could potentially express the gene products of that chromosome, were injected into mice, and then different mice would make an antibody response to the products of this constructed cell
Wow.
line. And then, the lab tested out, well, what are these antibodies reacting to? And one of the antibodies made in the mouse that was responsive to one of the chromosomes, I think it was chromosome 21, responded to a receptor, and the antibody blocked the interaction of interferon with this molecule. That was kind of the start of understanding what an interferon receptor might do. Yeah, so it turns out that there are two chains of the interferon receptor called IFNAR1 and IFNAR2. And I was particularly struck in reviewing, a lot of the literature that an investigator named Gideon Schreiber just did such detailed work on characterizing the relative affinity and effects of interferon beta versus alpha binding to the receptor. So one observation is that interferon beta has increased affinity compared to interferon alpha with IFNAR1. And, it might be important in how these two members of the type 1 interferon family behave in the context of cell response.
It's amazing the level of meticulousness that must have been involved in those experiments. So the interferon response also encompasses a unique form of signaling termed interferon-stimulated genes, or ISG, and interferon-stimulated response elements, or ISRE. Can you explain those concepts to our listeners?
Right. Well, I think some of this work just reflects how important focus on a particular pathway can be very, very broadly in medicine. So the work you're asking about is the work that's led to our JAK inhibitors that are used for many different diseases now. I think the key players in this part of the story are George Stark and Jim Darnell. And what was recognized was that exposure of a cell to type 1 interferon would generate a response that didn't really require new protein being made before the response was seen. So there was kind of a very quick connection between interferon, its receptor, and the cell responding with new mRNA, new proteins being made. And it was described over time that, hundreds, maybe 1,000 or so new gene products could be induced by exposure of a cell to type 1 interferon. The ISRE that you mention is a response element, a sequence in the regulatory region of a number of different genes, hundreds of genes, that could particularly respond to something triggered by type 1 interferon, although not interferon itself. So some interesting experiments were done where this ISRE sequence, regulatory sequence, could be placed in front of other genes, irrelevant genes, and they could be turned on when the cell was exposed to interferon. So hundreds of genes, interferon stimulated or interferon regulatory genes, or interferon regulated genes. And I think we still don't know what all of those gene products are doing, but many of them are part of the host response to viruses. And many of them may be contributing to some of the immune changes that we see in some of our diseases like lupus.
Yeah, so incredibly important. And I knew you'd get to the JAK-STAT pathway eventually. So do you mind explaining some of the interactions of interferon with the JAK-STAT pathway, and tell us a little bit about the tuning of type 1 interferon responses, including this suppressor of cytokine signaling, or SOX process?
So the interferon receptor had been described, and as I just talked about, the fact that interferon could turn on these hundreds of gene products was understood. But the question was, how does that happen? And I think Jim Darnell's lab working at Rockefeller gets a lot of the credit for dissecting the pathway, the interferon pathway downstream from the receptor. And what was found was that there are these proteins that bind to the intracellular part of the interferon receptor, and they came to be called JAKs and STATs. What's important and I think fascinating is that these JAKs and STATs, proteins that bind to the intracellular part of the receptor, are not just specific for type 1 interferon pathway, but they're really a kind of a rule for how cytokines across the board activate cells. So, with a lot of work over a number of years, the Darnell lab identified that TIC2, which is, even though it has a different name, it's a JAK family member, binds to IFNAR1, one of the chains of the interferon receptor. JAK1 binds IFNAR2. And there are some phosphorylation changes that go on when this happens. And then what happens next are these so-called STATs and become activated. And so it's kind of like a series of dominoes falling. Once the interferon hits its receptor, the JAKs become activated, STATs become activated, and then a complex of STAT1 and STAT2 with another so-called interferon regulatory factor moves to the nucleus, and it's that complex that turns on all of these interferon stimulated genes. So you asked about the suppressor of cytokine signaling molecules, and you would hope that a system that's so powerful and so important would be carefully regulated. And I think it usually is very, very carefully regulated. And one of the ways it's regulated is that among the interferon stimulated gene products are these proteins called SOX molecules, suppressors. And what they can do is associate with the JAK stat complex there and turn it down or inhibit its activation. So the interferon system has its kind of own built-in regulatory system. So at least a few of the genes that get turned on by interferon are geared to turn off the signaling process. And, hopefully that usually works okay.
Yeah, that's really a great explanation. So there's a little bit of a twist in the story now where interferon science intersects with the AIDS epidemic, which brings us to the clinician scientist Fred Siegel. Can you just share what he described?
Yes, well, we were all trying to understand back in about 1981, what was going on as all of these young men in New York were dying. I remember it well. I mean, that's the period when I was working at Rockefeller. And, I didn't really know Fred Siegel, but I kind of knew of him. He was someone who, I don't know if you or anyone who's listening know about Charlotte Cunningham Rundles. I think of her as kind of our current go-to clinical immunologist who we go to with immunodeficiency problems. Well, Fred Siegel was that person back in the early 1980s. And in fact, he was the first author on the New England Journal of Medicine paper in 1981 called Severe Acquired Immunodeficiency in Male Homosexuals. And so he was right there at the beginning of the AIDS epidemic and, was very involved in both understanding what was going on there and in taking care of patients in New York City. So he was a curious person and followed the immunology literature. And it was later in kind of the late 1990s that he was attending a lecture by a very excellent immunologist, investigator, molecular immunologist, Young Jun Liu. Dr. Liu had been characterizing these dendritic-like cells, which he was calling plasmacytoid dendritic cells, and trying to understand, what they were and what they were doing. I mean, going back a ways to 1983, Lars Ronblom, who I really consider kind of the father of studying interferon in lupus, had described what he called natural interferon producing cells. They were cells that weren't T cells, they weren't B cells, they weren't macrophages, but it really was not clear what they were. Well, in the late 1990s, Fred Siegel, this clinical immunologist, attended a lecture by Dr. Liu, who was describing his plasmacytoid dendritic cells. And Fred Siegel said, these cells smell like those natural interferon producing cells that have been described. And so Fred Siegel and Dr. Liu got together and they ended up publishing in 1999 a paper in Science with Fred Siegel, the first author, and Dr. Liu, the senior author, describing plasmacytoid dendritic cells as the key type 1 interferon producing cells. So, kind of a very nice bringing together of different expertise and different experience to really nail down what cells were really the robust producers of this cytokine family.
That's just a great part of the story. And I love these stories about kind of chance encounters between scientists. And I love that someone obviously so experienced and so knowledgeable, what we learn is that they still go to lectures that are touching on a topic that is
Yeah.
maybe a little outside their field. And that's where the kind of catalyst of collaboration can sometimes happen. So now there have been several clinical applications for interferon. Recombinant interferon therapy is being used in hematologic malignancies, treatment of hepatitis, and multiple sclerosis. Tell us a little bit about the science behind those uses.
Yeah, well, I mentioned that back in about 1980, there was this just intense competition to characterize interferon because it was believed, probably based on not very much data, that interferon could be a very important anti-cancer treatment. And so there was great effort to try to identify activity in different kinds of cancer. And, overall, I'll say that probably the hopes back then have not been realized, at least at the level that the excitement back in 1980 might have predicted. But particularly, say, in hairy cell leukemia, and as you mentioned, in a couple of autoimmune diseases like multiple sclerosis, type 1 interferon is given as a therapy. I think it's not the only therapy used in those settings. Hepatitis virus infection, as might make sense, is also an indication for use of recombinant type 1 interferon. So, yeah, there are a few situations in which type 1 interferon is used therapeutically to the benefit of patients with several different kinds of cancer and several autoimmune diseases.
But as with almost every component of the immune system, there's a balance, right? Where
Right.
too much can become pathogenic, timing of exposure seems to matter. And that brings us to lupus, another piece of the interferon story where there have been a number of twists and turns, several vital scientists, including yourself. So I think you report in your manuscript features reminiscent of autoimmunity were being described in those patients receiving interferon therapy for malignancy and other diseases. And as technology has advanced, the microarray gene expression studies in lupus have started to show elevated expression of interferon in the lupus population. Can you bring this all into context for us?
Yeah, well, the focus on interferon in lupus is also a very long story. And, I'll just briefly mention again an example of an investigator who didn't get too much credit for an important observation. And I have a personal aspect to this story because I was called by this investigator's son and asked, why don't you give my father more credit? It was kind of amusing. There's a paper published in the New England Journal in 1979. Hooks is the first author. And many of us over years that we've worked on interferon cite the Hooks paper as very important and recognize recognizing elevated levels of interferon in patients with lupus as well as several other autoimmune diseases. I mean, it turns out when I looked into that paper in more detail that it actually wasn't type 1 interferon that Hooks and his colleagues were reporting on. It was actually interferon gamma. And we haven't talked about interferon gamma, but
We haven't.
interferon gamma type 2 interferon is a completely different type of cytokine produced mainly by T cells and some other cells. But if you look in detail at that 1979 paper, they were really studying interferon gamma. And a couple of years later did confirm that, well, type 1 interferon is the interferon of particular interest in lupus patients, although interferon gamma can also be made. But it was Dr. Skirkovich, a Russian investigator's son, who called me. This was maybe, I don't know, 12 or 14 years ago, and said, as I said, you all are always citing that 1979 Hooks paper. My father recognized elevated levels of type 1 interferon in patients with several different autoimmune diseases. And I found these papers, and sure enough, Dr. Skirkovich even suggested that type 1 interferon could be a therapeutic target, which has come to pass. So, a lot of interest back in the 1970s, even in the possibility that type 1 interferon might be playing a role in autoimmune diseases. But as you mentioned, several of us took advantage of microarray technology, which was pretty new in around the year 2000, to look in the blood of lupus patients. And so there were several of us, several groups, particularly including that of Virginia Pasquale, who Virginia had already recognized that interferon alpha in the blood of lupus patients could promote augmented antigen presenting function by macrophages in lupus patients. So she was kind of on to interferon alpha having an important role. Tim Barron's lab, Emily Beschler and Tim Barron's lab, used microarray studies to identify interferon-induced genes, and then we did the same. I guess my recollection about that was that we had a poster in the 2002 ACR meeting, and Tim Behrens and Emily Beschler had a poster right next to ours. And, we both had those nice heat maps, red and green heat maps, which I think are a lot of the reason that we all kind of jumped on interferon at that time. It was so striking to just look at these heat maps with type 1 interferon-induced genes turned on in the lupus blood. And we were kind of debating, well, is this interferon alpha or is this interferon gamma? And, I was a strong believer that what we were seeing was type 1 interferon and interferon alpha. And at least my recollection is that Tim was not so sure, but I think we all came around by the following year to understand that type 1 interferon was having a big signal in the blood of lupus patients with many of these type 1 interferon inducible genes turned on. I'll just note, and I note in the review, that if we look back to 1988, there was a German investigator, von Wissow, who really made the same observation without the benefit of microarray. He found turn on of interferon inducible genes and interferon in the skin of nearly all lupus patients that he studied. So while the microarray and those red and green heat maps sent a really striking signal about the importance of this pathway, I think the observations were really out there if you looked for them.
Yeah, another highlight to the ACR poster hall.
Right.
Really a great place
Yes, we
to
definitely
collaborate.
need posters, right?
So why do we think that people with lupus have elevated type 1 interferon? What are the drivers? And what can we learn from patients with rare genetic syndromes to help us understand some of the nuances of the immune system in health and disease?
Well, we know a lot about the answer to your question, but not enough. So, one of our early observations. Really, it was a clinical serologic observation in studying our patients who had this so-called interferon signature, was that there was a very strong association between elevated interferon pathway activation and autoantibodies that target RNA binding proteins. So, as an example, anti-SM or anti-Rho or anti-RNP. And I think that that observation suggested that perhaps a response driven by an immune complex that contains RNA might be very important in driving the production of interferon. And I think that interpretation has been supported in many, many different ways by many investigators. I think the association of interferon pathway activation with the anti-RNA binding protein antibodies was very nicely supported in a study maybe 2 years ago from Peter Lipsky's group. Others are looking at the role of toll-like receptor 7, which senses RNA and other components of that pathway and turning on interferon. So I think a lot of the answer to the question is that if patients make that particular type autoantibody that binds an RNA binding protein and allows a complex to form that includes RNA and can access a toll-like receptor that senses RNA, that can be an important driver. But then why does that not turn off? I think that's an important question that we don't really have the answer to. And you asked about what we can learn from some of the genetic conditions where there are mutations in components of some of the important host defense pathways. There is this syndrome called Aicardi-Goutieres syndrome. And, we won't go into every detail about that syndrome, but it's observed in children with any one of a number of mutations in proteins that regulate intracellular nucleic acids. So some of the gene products will cleave up DNA or cleave up RNA or inhibit the signaling of intracellular DNA or RNA. And based on those mutations and thinking about their consequences, these children have very high levels of type 1 interferon. There is the thought that perhaps one of the reasons that type 1 interferon production is sustained in patients with lupus and some of the other autoimmune diseases is that there are some abnormalities in that pathway, the pathway that senses intracellular nucleic acids. And there are a lot of fascinating ideas about that and a lot of data that, might support that idea. For example, mitochondrial DNA might be one stimulus that, if not properly controlled, could turn on type 1 interferon. We've had this kind of fascination with genomic so-called retroelements, virus-like sequences in our genome. If those are not properly controlled, they might turn on type 1 interferon. So I think we have a lot of confidence that RNA triggering toll-like receptor 7 is a really important mechanism for inducing type 1 interferon, but a lot of other interesting ideas about additional mechanisms that might be going on, at least in some people.
Yeah, really fascinating. Lots to learn. So I wanted to wrap up the immunology part of this discussion by bringing things back to the clinic. We now have a monoclonal antibody that binds type 1 interferon receptor, and it's approved for lupus treatment in the US and I believe in Europe now, and that's anifrolumab. As someone who's worked in this interferon space for many years, are you willing to speculate on how interferon-directed therapies for lupus and other rheumatologic diseases might evolve?
Well, I can always speculate. I think that the type 1 interferon pathway and sustained expression of type 1 interferon over time, often years in lupus patients, is a really key part of the disease pathogenesis. But at the same time, there's clearly an essential role for altered B-cell differentiation and generation of autoantibodies. So both of these key aspects of disease are, I think, essential and will need to be controlled to have sustained therapeutic response. That's my view. I mean, how interferon can be best controlled, I think it's a work in progress. Blocking the interferon receptor is rational and good and I think is helping some patients, but it doesn't really get to the question of, can we turn off the production of interferon? It's just really blocking its downstream effects. So there are many other ideas, and we've talked about some of them. I mean, can we target the plasmacytoid dendritic cell that produces interferon? Can we understand more about those anti-RNA binding protein antibodies that might be driving the production of interferon? Can we block TLR7 as an important sensor of RNA? So a lot of ideas about how we might more effectively treat lupus, bearing in mind the importance of type 1 interferon. But I think at the same time, we have to be thinking about how can we modulate that B cell arm that generates the autoantibodies.
I love that you were even willing to speculate. It's such a complex process. So I want to thank you for that fabulous walk through the history of interferon and maybe take you down a slightly different pathway now. Our listeners love hearing about the people behind the papers. And as one of rheumatology's leading ladies, I wondered if you would share a little bit about your career. How did you get interested in immunology and lupus in particular? And were there any mentors who impacted you on your journey?
Oh, Vicky, it's a long story for sure. I had no plan to do what I'm doing, that's for sure. I ended up as a technician in an immunology lab after college and worked there under a very supportive mentor, Mark Wexler. He was a nephrologist and slash immunologist. And Mark gave me a lot of freedom in the lab and kind of crazily to kind of go off in different directions. And I guess I found it to be just a very supportive environment for learning about immunology. And during that period, I actually did encounter some of the lupus clinicians and started learning about what lupus was. Went to medical school and did all of the usual things there, and then ended up in a lab at Rockefeller, a really kind of intense lab, one of the few women there. But again, really loved trying to dig into how the immune system was working and how it might go awry in the autoimmune diseases. So somehow I just, the questions and the puzzles just kind of resonated with me, and they still do.
And you've been at the Hospital for Special Surgery in New York for most of your career. It's an incredible institution. What makes it special?
Well, it is a special hospital. And, as you mentioned in the introduction, I served as the physician in chief from 2010 to 2020. And that role, it's really overseeing all of the medical side of what goes on at the hospital with many, many rheumatologists, but also other medical specialties. It certainly gave me a great view of what does make Hospital for Special Surgery special. It's known mostly as an orthopedic hospital. And, we rheumatologists kind of fight back against that because we think that rheumatology is equally important here. But maybe from the point of view of how HSS takes care of patients, I've seen how it works as a system. And maybe this is a lesson for, our healthcare system in general, or, the hospitals where we work. HSS has excellent rheumatologists, it has excellent surgeons, but it's put together such a kind of cohesive way of taking care of patients, whether it's, how the patient experiences being in the recovery room, all of the medical evaluation before surgery and after surgery. And that's a role that many of our rheumatologists are very much involved in leading. Just the whole picture is well done and coordinated. So, that's at least a part of the answer. I think from the research side, we are very, very interested, and maybe I'm an example of that, of getting to the bottom of disease pathogenesis. We don't want to just treat rheumatoid arthritis. We really want to understand it. And certainly the same has been true of lupus over many decades. So yeah, I've never really had any urge to leave HSS. It's been a great place to have a career.
Oh, wonderful. And tell us a little bit about how you got involved in leadership, both in your own institution and then also in the field of rheumatology. And do you have any advice for other rheumatologists aspiring to leadership roles?
I don't know. That question might be difficult for me. I feel like I've never really pushed very hard for leadership, but at the same time, I do something that people are instructed not to do. I tend to say yes to things. And I do it just not because I forget to say no, but I really feel like the way we learn and make things better in general is by being willing to be involved in things. So I became the physician in chief here after, a search had been going on for a while and I wasn't really paying attention to it. And my predecessor, Steve Paget, said, why don't you apply for the job? And I guess I couldn't think of a reason why not. For ACR, I remember, this is years and years ago, going to a dinner with Mark Andrzejewski, the former administrative leader of the ACR, where it was almost like a recruitment dinner. And somebody must have told him, maybe Peggy Crow would be somebody to get involved in some of the ACR committees. And so he suggested I join a committee. So that's kind of the way it happens. And I guess what it is, I get pleasure out of working on whatever it is to try to make it better. And, maybe part of that is my own difficulty in just depending on myself to kind of advance my own interests. I don't know. I have some discomfort with that. So anyway, getting back to your question, how might we advise our colleagues who want to develop leadership? I think, don't say no too often and think about saying yes, because every experience you learn from and you become a little bit better at being a leader.
I think that's great advice, actually. And one of the points that really struck me when I was reading your manuscript is that life in science has many dead ends, wrong turns, things that at first glance seem to be failures and then suddenly turn out to be turning points. How have you navigated some of that as a physician scientist?
Oh, well, I guess I feel like I've probably never with regard to my own interest, pushed as hard as I could have pushed to, follow every lead, complete every data set as much as I should. And it's, maybe a deficiency of mine. But at the same time, I haven't really shut down pathways or areas of interest that aren't working. I have probably too much data that's just kind of sitting around that I should get back to and think about. So I don't know. I guess my tendency is to really value data, try to collect data in a good way. And, my own lab is very much focused on following patients longitudinally, both from the clinical point of view and from the biologic point of view, and then kind of look in that data for really interesting and maybe significant leads. So there are probably many pathways that haven't been very successful, among all the things I've thought about. But I guess the answer is just to keep an open mind, try to connect dots, and try to focus in on what looks the most promising and not worry about what might be less promising.
Yeah, great advice. I really want to thank you for joining us on the show today. I know many rheumatologists will be grateful for you sharing this excellent history of interferon, but I think most of our audience will also really appreciate your insights into life in science.
Thank you, Vicki.
Our thanks today also go to everyone who's laid the groundwork for the immunology we practice at the bedside. One of the fantastic things about science is that it's both communal and iterative, and the interferon story exemplifies that. But as scientists toil at the bench and rheumatologists toil in the bedside, we should never forget that sometimes discoveries are only appreciated looking back. The arc of history is long, and for much of rheumatology, that narrative is still being written. Thank you all for listening to this podcast. ACR Journals on Air is a production of the American College of Rheumatology. Please follow at AC Room on social media. Email us at podcast@rheumatology.org and rate and subscribe to our podcast at acrjournalsonair.org or wherever you get your podcasts.